Good morning, everyone. Happy Friday. Thank you for joining us for today's Team Research Forum that we will be focused on research looking into our preparation for the next pandemic. It's just 10:31, and I know there's a lot of people joining us from either teaching or earlier meetings, so we're going to wait a couple of minutes and give everyone a chance to join us. We ask that if you're not a speaker, you keep your microphone muted and your camera off, and that will help with bandwidth. But if you do see a colleague in the participant list, please feel free to send a message or say hi, and we'll get started in a couple of more minutes. Thank you. Just reminding everyone again to please mute your microphones. It's 10:33, so let's just get started then. I'm sure we'll have more people joining us in the next few minutes. Again, welcome to the Team Research Forum this morning. We have a very exciting panel that will talk to us about research and activities on our campus looking into preparing for the next pandemic. This panel brings together specialists working across a range of fields, working on catalyzing innovation for rapid identification of pandemic threats through advanced biosensors, field-forward surveillance in remote and rugged settings where spillover is most likely to emerge, and modern information science and data analytics that can advance predictive science. It is my pleasure to introduce to you the leader of this team, Professor Christine Johnson, who is the director of the Epicenter for Disease Dynamics, and is a professor at the School of Veterinary Medicine. Dr. Johnson's research looks into to understand the impacts of environmental change on health, and she has guided surveillance activities at the interface of animal and human health for emerging pandemic threats in Asia, Africa, and Latin America. She's particularly interested in focusing characterizing fault lines for disease emergence, spillover, and spread. And findings from her research are designed to inform public policy and better prepare for emerging threats. In this panel, she is being joined by Professor Christina Davis, our recently joining Office of Research as Associate Vice Chancellor for Interdisciplinary Research and Strategic Initiatives. We have Professor Jonathan Eisenhower from the Department of Evolution and Ecology. We have Professor Xiaodan Kong, Associate Professor at the Department of Mechanical and Aerospace Engineering. Professor Pranav Pandit from the Epicenter for Disease Dynamics, and Assistant Researcher Epidemiologist Chiara Evans from the One Health Institute. Our panelists are going to do a short presentation for us to give us an overview of the work going on in this very strategic area. And after that, we will go ahead and start with the QA session. So I'm going to pass it on to Professor Johnson to get us started with the presentation. Thank you.
Thank you so much, Ana Lucia. We're so excited to talk to you all about what we've been working on together. This is the panel, as Ana Lucia said. I wanted to kick us off by saying Happy Earth Day, everyone. This is actually the perfect time for us to think about pandemic risk. We think that our pandemic risk is very closely tied to the health of the planet. So this is, I think, very appropriate for today. So this is some data that's been published recently that shows how the risk of large-scale epidemics has been changing over time, specifically epidemics that are caused by emergence of novel viruses. And you can see here that the trends really start to emerge in the 1950s. So there was, of course, the Spanish flu, the Great Influenza in 1918. And then starting in the 50s, we have this sort of trajectory where we see quite often a new virus emerge that causes an epidemic in humans. And we think that these trends are not unrelated, let's just say, to the similar trends in population growth. So there has been, of course, a massive trajectory for increase in human population, especially in some of the tropical regions where a lot of these pathogens have emerged. Here's about the 1950s on this graphic that shows you since BC, sort of the massive increase. And we think that the increase in the scale of the epidemics is largely driven by population density. So that makes so much sense from a disease dynamics perspective. There's also social and economic inequalities that allow diseases to take hold in populations and gain traction before they're recognized. And of course, trade and travel is going to increase the sort of breadth of any epidemic in terms of its trajectory across the whole human population. But we also think that the frequency of emerging infectious disease is driven by landscape change, specifically landscape change where people come into closer contact with animals around agricultural production and development, specifically for human dwellings. And so this is sort of the way that we are thinking about and wanting to investigate pandemic risk. We worry about deforestation, habitat loss and fragmentation, and the implications that then has for movement of wildlife to specifically accommodate that landscape change. And oftentimes, the way the landscape has changed, this invariably brings the wild animals into closer contact with humans. Those animal-human interactions pose the risk for emerging infectious diseases, specifically again from wildlife, because the vast majority of emerging infections have wildlife as their root source before transmission to humans. And so our team has been thinking about ways to ensure pandemic preparedness for a long time, starting from the last over a decade, actually, with the USAID Emerging Pandemic Threats Predict project, and more recently as part of this NIAID Center, the Epicenter for EID Intelligence, where we have looked at these research priorities fundamentally for us in this sort of pre-emergence and spillover and early emergence phase. So we're very focused on viruses with pandemic potential. We like to work where they're most likely to emerge. So that oftentimes is tropical regions in other countries, but certainly front and center in the US is, of course, SARS-CoV-2 and the risk that that has for transmission from humans to animals and back to potentially humans in the ongoing coming years of this current pandemic. And so this is largely informed what we consider the big gaps in our work going forward. And so we have been so excited to be able to work together with this whole panel that's presenting right here, as well as some external colleagues and Dr. Simon Anthony, who is also at UC Davis, around this new NSF initiative, which actually started for some of us in 2020, where we were selected by NSF, and it was very, I think, exciting for us because we were able to work across the 4 NSF directorates with the NSF directorates, in particular around biology, engineering, computer science, computer and information science, as well as social, behavioral, and economic sciences. And we hosted this workshop to initiate sort of a scientific discussion around the challenge of what they call the PIPP, or the Predictive Intelligence for Pandemic Prevention. And as far as our engagement with this panel here and our colleagues at other universities, that has largely focused on these big gaps that we have, or as NSF wanted to call it, these big grand challenges on how do we develop actionable intelligence. And so for us, it became very quickly focused on what impedes our fieldwork, what impedes our ability to do detection in that very early emergence phase. And so that is what we're going to hear from the rest of the panelists. We're looking towards these autonomous networks and new sensor technology, which was really exciting from the engineering side, as well as data systems and modeling coming from the more computational side of things. And then it's not lost on us that we really need to figure out how to work together across a breadth of disciplines that we hadn't really done before. So this transdisciplinary collaboration needs in itself, I think, a grand challenge effort and ways for us to guide each other so that we're communicating and enabling cross-disciplinary research in a really new way. So this is what we put together as part of our proposal. We put in a sort of a phase 1 towards an NSF center, and we're looking in this sort of post-COVID early emergence phase where there's ongoing transmission in humans, and then we've got transmission or maintenance in potential animal reservoirs of all kinds still yet to be determined, and what does that mean for potential spillover to humans. So this was very much built around SARS-CoV-2 in the US, and then other variants and other emerging infectious diseases elsewhere. And here's all the new technology and where we see that playing a major role. And this is just one example. You'll hear of a lot of other case studies from the rest of the panelists, but I just wanted to turn it over with the idea that we sample in all kinds of places that we think are really important for us to be able to continue to monitor and be able to do detection. We very much believe in sort of the integration of animal and human health. And so even when we're working in what would appear to be relatively remote settings, we actually have engaged the human community around those settings. We understand sort of the human use of that setting and are working with the medical doctors in that area to do early detection on the human side, as well as hopefully with this team here, really pushing the envelope as far as technology around some of the settings where we have, caves emerging from bats. Actually, this is some of the easiest places where we sample because at least the bats come out all together all at once. And we just have to put a net over the cave and then we can catch a whole bunch of them. It's not always that easy. There's bats of all kinds that roost in trees that we have a lot harder time. But just opening up the window, I think, for us to think about what are some of the ways that we could partner with our colleagues that can enable better characterization of these caves or access where we just simply can't fit into the opening, or other ways to sample, including around what could be considered sort of this effluent coming out of caves. That's quite common, something we see often. So with that, I'm going to turn it over to my colleague, Dr. Christina Davis. Thank you.
Great. Thank you, Christine, so much. Next slide, please. So one of the things that my group has been working on is looking at health relevant information that is in exhaled breath. When we breathe, we inhale oxygen, we exhale carbon dioxide, but we also exhale a lot of other content that's metabolites at both the volatile and non-volatile compounds. And we can non-invasively sample these and measure them if we have correctly engineered systems. Next slide, please. And so we have a paradigm where if you know some of the chemicals that you're like to measure and you know that they're significant for health, you can design sampling and instrumentation systems that can sniff those potentially. Volatile organic compounds are one of the categories of compounds that we look at and we measure frequently. We usually do some type of advanced data analytics, and then we have some way to have a system output that can tell us, red light, we detect something that is indicative of infection, and then green light, everything looks okay. Yellow light, maybe take another sample. So we have these system level approaches to sample breath, measure content in it, and then interpret the result. Next slide, please. And you may have heard in the popular press over the last 2 years, there's been a lot of attention looking at animals that have really good senses of smell, especially dogs, canine systems, where they can actually be trained to detect COVID-19 smells from human subjects. And we don't know if that's coming from the breath or if it's coming from skin, but whole body odor is something that they can be trained against. And there's a lot of different areas where these dogs are now being deployed. And from all accounts, they seem to do a really good job, but it's limiting in the sense that it's not easily scalable. And so we look and we continue to seek engineering solutions that help to mimic this and that can be deployed widely across different areas. Next slide, please. And one of the things that we are currently doing is I have some collaborators in the School of Medicine. Nick Kenyon in Pulmonary and Critical Care and Sleep Medicine is a collaborator on a new NIH U18 center grant that we have where we're looking at sampling human breath from COVID positive patients and then building libraries of the content that we find in their breath, that's metabolite content, and then looking at the markers of significance. And so patients, the human subjects would blow up a Tedlar bag, which is shown on the left-hand side. We then use a sampling device that's shown on the far right-hand side to suck the breath that's in the bag onto a sorbent bed, which is in that little middle picture. And then it sequesters all of the breath metabolites onto that sorbent, and we can take it to a mass spectrometer for analysis, and we can detect the compounds that are in it. Next slide, please. Next slide, please. And one of the things that we've been able to see is when we look at the total content that's in the positive and the negative samples, that we can very clearly distinguish one group from another. And from individual subjects, you can kind of see how their breath signature compares with that infected or not infected pattern. This is a study that's still in progress. We're still collecting data, but so far we're getting sensitivity and specificities that are, reasonably accurate. And as a layered strategy for public health approaches to detection of COVID, this is not one additional tool that we have in our arsenal, in addition to PCR tests that are the gold standard, as well as over-the-counter at-home rapid antigen tests. So we have new tools that are coming online to detect these emerging infectious diseases in the human host response to infection. Next slide. Well, we've done some work previously with animal systems as well. Several years ago, we were able to work with the U.S. Navy Marine Mammal Program, where we looked at breath volatiles that are in cetaceans. And cetaceans are an interesting group because they are also a zoonotic reservoir of disease. And so they can pass infections, respiratory and viral infections, back and forth between humans. And we, again, were able to develop tools. In the far right-hand side, you can see a device where we're actually placing this directly above the blowhole for the dolphin, and we're able to collect their breath and then retrieve it and sample it so that we can understand that metabolomic content. Next slide, please. And we can also do this in field use. This makes it very useful to take out into deployments, to do this in outdoor environments. And in this case, with the Marine Mammal Program, these dolphins are actually very used to working with humans, and so we could take samples from them pretty directly. But we also use this to do some field testing with samples from a wild population. Next slide. And one of the populations that we had access to was in Sarasota, Florida, where they actually do wild capture and release health estimates of the population. We were able to go along with a group that did that work and to take breath measurements from those animals non-invasively, which is a very good quality to have when you're working with marine mammals. And then we also were able to travel to Barataria Bay, which was inside of the BP oil spill area, which was in outside of the coastal area of Louisiana. And what's very interesting from this data is that the marine mammal animals in San Diego have a very tight cluster of the metabolites that we measure in their breath. They eat the same food every day, they have the same activity, they're in the same environment, and it's pretty tightly clustered. We see in the wild population in Sarasota, we see that those were spread out a little bit more. We see some diversity in those metabolites, but they still roughly clumped with the San Diego group, which is what you would expect because these are all the same bottlenose dolphin species. What was challenging about the Barataria Bay animals is that they actually were impacted heavily from the BP oil spill exposure zone. And according to the data that we took, as well as we did sonograph looking at lung and abnormalities, we looked at CBC blood counts, other tests, and endocrine tests, is that we saw that those animals were very ill. And what we found is in our samples in the breath condensate, we saw chronic inflammation metabolites that were also highly associated with that evidence of lung consolidation, which is, you know, which is something that would show up, and they were very ill. Next slide, please. So we then went further with that and looked at some animals that were under managed human care at San Diego. And we actually took longitudinal samples over time from the same animals. And what you can see here is the personalized responses of some of these metabolites that we measured in cases where they were healthy in green, and in cases in red where they had some type of condition. It could be, in this case, an animal that was pregnant or that had hormone alterations, but in this case, it was a pregnant animal. And you can see that they change over the time course, and that this is an adequate way to look at a personalized response for individual animals. And we think that the big picture of this is that you can use these measurements to kind of look at the health of the ocean, not just the animals, but the populations. Next slide, please. Now, switching gears a little bit, I talked about VOCs that are in exhaled breath from both humans and animals, but there's also a lot of volatile organic compounds that you can measure environmentally, and they can come from animals, plants, or other species, but they can also be metabolites of interest or compounds of interest that are in that environment. We developed a small chip-based sampler that you can see on the left-hand side. It has a small sorbent packed inside of it. And using this type of device, we can capture volatile compounds from environmental samples. We can return them to a base point, and we can measure them either in the field or back in the laboratory. Next slide. And we've done this with humans as well. So we can take these types of samplers. They're small, they're portable. You can attach them to people and they can walk around with them. You could, in theory, do this with other forms of remote sensing as well. And we were able to, next slide, we were able to detect the presence of volatiles as people moved around geographic areas. We actually timestamp, and we can geolocate all of the time points where the samples are taken. And you can not only put these on people, but you can also put them onto UAVs. And we've done a little bit of work with that, where we can fly these types of samplers around. We can even hook them up to real-time sensors and to be able to measure these volatiles in different environments. Next slide, please. And the last thing I'll say before I pass this off is, this could be very interesting in enclosed environments where there's effluent that's coming from different animals or species of interest, like urine, scat, or other sample types. Being able to measure the volatiles that are in those areas are another non-invasive way to take data from those environments. With that, I'll pass it off to my colleague, JiaBin Huang.
Yeah. Thanks, Christina. So what I'm going to do is I'm going to talk very briefly about using robotics and AI to characterize and sample wildlife reservoirs in remote settings. And then for the sake of discussion, so what I'm going to do is I'm going to use investigating bat caves as an example. So here you can see two pictures I took. Both of them, they are bat caves in Texas. And then if you think about the way that, let's say that we want to investigate these bat caves, and the way that we are doing right now, as I was told, that the scientists have to wear protective gears, and then have to get into these caves in order to take these samples. And then as you can imagine, this is very dirty and then very dull process, and then it can get very dangerous in the sense that, so basically this bat cave is very dark, and then some of these bat caves are very hard for humans to navigate. And then I would argue that this is a perfect scenario for us to use robotics to help us to explore and also to investigate these bat caves. Next slide, please. So this is what I have in mind. So what we can do is we can send a drone equipped with a LiDAR or LiDAR sensor. And then with some very standard technique, which is called SLAM, simultaneous localization and mapping, and then we can easily build a 3D map of the cave. And next slide, please. And then after that, once we have a 3D map of the cave, and then we can send a second drone. And now this drone is equipped with thermal cameras, which we can use it to basically to see in the dark, and also to be able to detect these animals, because normally their temperature, the body temperature is kind of like it's warmer than the surrounding environment, and you can see the animals. And then we can also equip it with the chemical sensors that Professor Davis' lab has already developed or is going to develop in the future. And then what we can do is similar to what Christina has done, and then we can time step the location of the drone with the information that we have from the sensors about either the density of the animals or the density of the VOC compound, and then we can superimpose that information to the 3D map that we have generated. And then this type of maps, basically the 3D map of the cave and also the maps of animal densities and the VOC densities, we can almost generate them in real time. And after that, what we can do is we can, let's say, that give these maps to our scientists, and then they can help us, or they can do them to figure out basically what are some of the hotspots or potential candidate set for further investigation. And then next slide, please. And we can send the third drone. Now this drone is equipped with a manipulator, and then it can be used to, for instance, to physically sampling either kind of urines or feces, and then they can bring these samples back to a field station and then for analysis. And after that, we are going to have a map of the cave, the animal densities, VOC densities, and even the pathogen densities, and so on and so forth. And then I believe this type of information will be extremely valuable for our scientists to kind of to study these virus spreading and stuff like that. Next slide, please. So personally, I'm an engineer, and then I believe that the role of our engineers in this endeavor is mostly to help our scientists to get high quality and sufficient data for them to perform scientific discovery. But in the meantime, there are a couple of very interesting engineering challenges that I think we need to address in order for us to do that. And the first one is, it is impossible or very hard for a human to remotely control a drone. The human is sitting outside of the cave, and then remotely control a drone which is flying inside. The reason for that is there is no Wi-Fi in the cave, and then radio doesn't work. And then ideally, therefore, you will need the drone to be able to navigate and then even to map the cave by itself. And then this is a very challenging problem. And then the video that you're going to see on the right-hand side is Christine, can you play the video? Okay, so something that we have done recently that in our case, basically, by the way, this is from the drone's perspective. So what we have done here is we use some machine learning techniques, and we're able to allow the drone to fly by itself without any human intervention, and then without a map of the environment, and without even GPS information. So basically, the drone is able to fly by itself by just relying on the onboard sensors, mainly a binocular camera. And another engineering challenge is how to track these moving animals. And then can you go to the next slide, please? So basically, the animals, they do not stay in the same place. They migrate, and then they emerge. They basically forage, and then they may interact with some other animals and humans. And then this is basically some of the work that we have done a couple of years ago. And then as you can see, this was taken by a summer camera, and then probably you can see the drone here. And then in this project, so what we have done here is we are able to use a drone to basically to track a swarm of bats emerging out of their caves. And then there are some other challenges, including, for instance, how to perform error manipulation and how to solve kind of swarming and human problem, in the sense that basically instead of sending one drone at a time, is there a way that we can send multiple of them together so that we can get this much more quickly. And for the sake of time, I'm not going to talk about the other two challenges. And then I would love to discuss this either today or offline about these challenges. Next slide. So, and then I'm going to pass it to my colleague, Chiara Evans.
Thank you. I'm going to speak briefly about non-invasive techniques for sampling wildlife, and I'm going to kind of switch topics here and focus on non-human primates. So one of the reasons we think that non-human primates are really important in pandemic surveillance is that they often really flourish at this sort of transitional ecosystem zone between forest landscapes and urban landscapes. There's the opportunity for contact with humans, and the other is that there's opportunities for more diverse species mixing, and we believe greater opportunity for higher viral host plasticity. So monitoring these sorts of ecosystems are really important. And for primates, there's kind of two different sets of challenges we have. So we have habituated primates that are really used to being in and among people and can often come quite close. And then you have totally unhabituated primates that may live really deep in the forest and may occasionally come to the forest edge. And those are really interesting because they can sometimes act as like a bridge population where their viruses that are circulating deep in the forest may be able to come to the edge where there are people. Next slide, please. So one of the important things to emphasize with non-human primate sampling is that understanding behaviors of the species and also behaviors of the individual populations are really essential. So for pandemic scale surveillance, we're going to require community-led monitoring. So we need to be able to work with the populations of people that live and see these wildlife all the time. So that's going to require innovation in field forward advances in data and sample collection. So working towards making the sample collection as simple and as possible in the field side. So just a few of the things we've been working on in various projects have been smartphone data harnessing technologies and citizen science. How can we get local populations to report information about primates on their basic phones or their flip phones or smartphones, information about diseases or information about movements, and also how can we simplify biological monitoring of primates. So one of the most important things to consider is how can we get rid of that pesky cold chain requirement. Most of the places we work, it's virtually impossible to get electricity, and sometimes even getting dry ice or liquid nitrogen is really, really challenging. So here on the right here is just an example of one of our field sites where we're working with elephant loggers that can go much deeper into the forest than we ever could. And they're working with microsampling mechanisms to collect small drops of blood from freshly killed bushmeat from primates that are brought into villages or into logging communities. And we're able to look for antibodies to pathogens against using those sample types. And then we've also been working on ways to optimize the collection and testing of wildlife byproducts, so things like feces and saliva from primates. Next slide, please. So two of the techniques we've worked on for collecting non-invasive saliva collection, one is just the very simple use of dropped chewed food. So on the right there, you can see a mountain gorilla. They spend most of their days chewing and sort of spitting out pieces of dropped forage food. And we can observe that and pick those samples up and actually detect viruses off of the saliva from those pieces of chewed plants. The other method we've worked with is the distribution of sort of little chew ropes with an attractant on it. And they'll chew those little chew ropes kind of like bubble gum and then spit it out. And then we can centrifuge that and get a really nice saliva sample. And both of these methods have worked well for detecting RNA and DNA viruses. However, more recently, we've been trying to optimize them for the detection of antibodies against arboviruses. And we've successfully been able to detect antibodies against Japanese encephalitis virus, Zika virus, and chikungunya virus in non-invasively collected primate saliva samples using the MagPix platform that we have been collaborating with at the California National Primate Research Center. Next slide, please. Oh, back one slide. There you go. So just to kind of re-emphasize the importance of understanding the primate behavior. So these are various examples of different populations we've used these methods with around the world. In the top left here, you have some sanctuary primates in Peru. Below there, they have rhesus macaques in the very urban center in Nepal, where these were very used to being close to humans. And we actually had to adapt some of our methods because they were getting too close to us. Then we have Lahostes monkeys, red-tailed guenons, and olive baboons that are kind of semi-habituated in Uganda. And then we have mountain gorillas from Rwanda on the right there that had to be monitored completely non-invasively using dropped chewed plants. Next slide, please. And then I just wanted to share a couple videos. We'll do the one on the left, shows the differences and challenges. So this was a particularly wonderful sampling day. The entire group of mountain gorillas actually left the park and was in a wide open area where they were eating eucalyptus trees from villagers' crops. And we were able to sample the entire population, the entire group in that day in a matter of an hour, because it was quite easy. And then on the right here, the right video, if we move over to that one, this is taken just a couple weeks ago. This is on the border of Myanmar and China. You can see way up there, that is a hulat gibbon. And this species is not at all habituated, this population, to humans. And so the only time they ever really encounter humans is when they're being potentially hunted. So as you can imagine, the first sight of us, they're gonna run straight back into the forest. So for this group, we are relying entirely on small pieces of chewed plants that they drop from a really high elevation. They very rarely come down to the forest floor during their daily activities. Next slide, please. While it loads. Here we go. So just to sum up sort of this just brief discussion here on non-human primates, some of the things I hope we're able to focus on for future innovations in less invasive primate sampling is taking both a broad and also an in-depth approach. So we want to be able to increase the numbers and types of species that we're able to collect samples from non-invasively, but also importantly, we want to be able to increase our ability to go back to a population when we found something really interesting and follow them longitudinally and repeatedly over time to understand transmission dynamics for whatever pathogen of interest we found. And that absolutely is going to require more innovation in non-invasive and remote sampling, because we need to be as hands-off as possible so that they'll allow us to engage with them more frequently over time. So one of the things I think would be really exciting to work on would be saliva monitoring stations. So how can we get something stationary that we can leave ideally high up in those treetops where those gibbons are, that will allow us to collect a saliva sample without any possible interaction with people, so they don't even see us. Also, how can we improve things like remote biosamplers using mosquitoes or ticks or other vectors that may be feeding on these primates that may be easier to collect than, say, the actual primate themselves? And can we look at the blood meals in those vectors and get information about pathogens that are circulating? There's also been quite a lot of innovation in swallowed biosensors in human medicine in the last few years. And I think there's a lot of opportunities for us to think about transitioning some of those technologies to some of the non-habituated primate populations. And also, as we're moving towards really requiring much smaller sample volumes for some of these diagnostic testing platforms, we really don't necessarily need a whole volume of blood that we are used to collecting. We can probably get away with 5, 10 microliters of samples going forward. So I think it's time that we can start thinking about how do we make our darting mechanisms less invasive? Can we use darting mechanisms that collect a very small piece of tissue or even a drop of blood that allows us to get more information than we were previously? So I think there's a lot of exciting things that we can work on to improve making all of our field work much easier. And next, I'd like to turn it over to Dr. Pranav Pandit.
Thank you, Dr. Chiara Evans. I'm gonna start sharing the screen from my side. With my talk, I'm going to talk about the computational challenges and opportunities that we have for pandemic preparedness and developing intelligence about emerging viruses and emerging pathogens from wildlife. So if we look at multiple ecological mechanisms and barriers that these pathogens face before they spill over into humans, they can be kind of classified into 3 broad categories. The first one, as described in previous slides, is when these pathogens are just transmitting and circulating within wildlife populations, followed by the next one where they are interacting with humans. These wild animals are interacting with humans and provide opportunities for pathogens to transmit into humans. And finally, once they are established well into human populations, those transitions also provide us some opportunities for developing some intelligence and developing some mitigation strategies around those. When we think about these stages from the perspective of the pathogen, we can think about its evolutionary trajectories, starting from its circulation in the wildlife reservoirs to some stages where it's causing short outbreaks in human populations, and then eventually sustained transmission within human populations, where it is just transmitting between humans, and there is no requirement for further spillover to happen for as a requirement for human cases. So during my small presentation, what I'm going to do is kind of show you a few examples of some models that we have been developing to have some intelligence around these barriers. Develop some predictive models that we have been doing based on some of the data that others have been collecting over the last few years. So the first thing I'm going to talk about is about developing models that can help us develop surveillance, which is very much targeted for emerging zoonotic pathogens. This is to understand and predict regions with high diversity of wildlife hosts, and hence have high vulnerability for zoonotic transmission in human populations. Some of this work was developed during the earlier pandemic threat that we faced of Zika virus. So we started focusing about wildlife reservoirs of flaviviruses, a larger group of viruses, which also includes the Zika virus. And we were able to develop some models that can help us predict missing hosts or hosts that we are yet to observe for all these flaviviruses. And we were able to identify regions where there is a high diversity of these wildlife hosts that can harbor these flaviviruses eventually. And these have been helping us develop a targeted surveillance for multiple flaviviruses, including yellow fever and Zika, West Nile, St. Louis, dengue, Japanese encephalitis. And what's really useful of these models is that previously, before these modeling exercises, we knew very little about the possible hosts that these flaviviruses can infect. But now we have a list of hosts that we can target to understand their ecology much better. For example, now we know that to understand the ecology of yellow fever and Zika virus, we probably need to focus more on primate hosts that are in those regions. For Japanese encephalitis, there is a diverse host species from passerines that we need to focus on. And then for dengue virus, along with the known hosts that are primates, we also need to focus a lot on bats, especially New World bats in South America. Going ahead, the next system that I'm going to briefly talk about is to develop some surveillance intelligence based so that we can understand and have early detection of wildlife health events that are happening in wild population. And that's really important because these unusual wildlife health events help us understand or predict if there is any increased pathogen release happening from these wildlife hosts, which can not just be of concern for wild populations, but can also have public health consequences. And to do that, we were kind of able to set up a surveillance system here in California, which is based on the rehabilitation centers that are at the forefront of handling wildlife cases. And we were able to establish a new surveillance system, which is online and everyone can easily access it now as well, called as Wildlife Morbidity and Mortality Event Alert System. So you can go into the system, create your own account, and set up surveillance targets of your own interest. For example, here in the picture, you can see there is a target set up for mammals with dermatological diseases. So in the back end, we have been developing multiple machine learning models that standardize all the data coming in from these rehab centers. It's it's crunching numbers to understand what's an anomalous behavior in those cases, and generating alerts when there is an unusual number of cases that are being reported for certain targets. So just for an example, we can think about neurological diseases happening in birds, and whenever there is an alert or unusual number of cases, the system will let us know, let the user know that there is something unusual happening. So it is already generating a real-time intelligence for wildlife health, which is also significantly important for public health for many of the emerging viruses that also infect wildlife. Thirdly, I'm going to briefly also talk about methods that we are trying to develop here at the Epicenter for Disease Dynamics to understand what are the risk of zoonotic transmission for novel viruses. Many times we do find these novel viruses in wildlife, but we haven't yet found them infecting human beings. So we really need to start quantifying the risk that these viruses pose to human beings. So we are trying to understand what is the evolutionary trajectory of these novel viruses that are currently just transmitting within these animal populations to eventually come into stages, the later stages of the evolutionary trajectory of the spillover. So we were able to develop some models that are based on networks of known zoonotic and non-zoonotic viruses and their hosts, including many of them since many of them are zoonotic, so they do have humans as their host. And we were able to generate predictions for 531 novel viruses that are only detected in wildlife, and develop a prioritization score metric system that kind of gives score to these viruses, whether they pose any risk for human transmission in the future or not. And this is just showing how the prioritization score behaves when it comes to known non-zoonotic and zoonotic viruses. So you can clearly see that there is a higher chance, higher prioritization score for known zoonotic viruses. And some of these predictions, I'm just showing it for the family, Coronaviridae family, coronaviruses, the novel coronaviruses. You can see over here and how their prioritization scores stack up against each other. There are top 10 coronaviruses that are in this plot versus the bottom 5, which has a very low prioritization score. So how can we use these matrices is the way we think about this is that these can help us prioritize some of these viruses for further studies, especially people who are doing in vitro studies, in vivo studies, and then eventually also help develop some additional ecological data around these specific viruses by doing additional surveillance in species in which we have found those viruses already. So it is kind of, again, helping us develop targeted surveillance for these possible threats for public health. So what are the future challenges and opportunities around these issues? When it comes to specifically toward challenges, the way we think about is that there is a lot of challenges around data that we put into these models. The first and foremost important one that we always face is around the surveillance bias that we see when we are trying to do global models, like the one I just showed for flaviviruses, where there is so much of difference in the efforts that are put into sampling or surveillance data collection that has happened over the years across the regions and across different viruses, and accounting for those can be a difficult challenge for many of these models. Along with that, I think availability and integration of ecological, molecular, and anthropological data, which all of those play an important role in these models, is another important challenge. When it comes to modeling frameworks, the key challenges that we face are around the scale of available data. Most of our data is at individual level data, where we are trying to sample those animals and understand whether it's positive or negative, while the modeling frameworks many a times can be at a different scale, at a population scale, or at even a regional scale, which can be a hard challenge to account for those differences. There is definitely some methodological challenges that we are working on, especially to develop new algorithms that can help us understand anomalous trends in these surveillance data, especially for emerging pathogens. So, like for example, if there are fewer cases of certain new virus that we are looking that we are detecting, let's say either in humans or in wild species, what is the level at which we really need to trigger an alert or be more concerned about those cases? I think all those methods need to be developed really well so that we can keep generating those alerts and generating those intelligence around the data that we are collecting. I think it just stopped sharing.
Yep. Sorry, Pranav, we better turn it over to Dr. Jonathan Eisen.
Perfect. Yes, that was my last slide. Thank you.
Perfect. Yep.
Yeah.
Great.
All right, I'll try to be really quick here. I know you're running behind. So I'm going to take a step back. I switched the order of my slides here to take a step further back, which is I want to convince you that we can think of what we're doing here in a way much like people study birds. So if you are familiar with this, it's a normal bird. If you knew what it was, you would know it's boring and uninteresting. It's an American robin. Seems pretty normal, right? Many years ago, maybe 15 years ago or so, people observed an American robin and they freaked out. Why did they freak out? If you can see, the cars are parked on the wrong side of the road here. That was in the United Kingdom, and they didn't expect to see an American robin there. As I've told people this before, unfortunately, they also watched as a falcon ate the American robin while it was in England, but they got very excited. And the birding community is a really good example of how certain groups of people with certain types of information can identify something as abnormal really, really quickly and integrate it with information. And the way they do this is they have basically field guides where they know where organisms are supposed to be, what different types of organisms are out there, how to identify those organisms, and then, you know, can build maps of information from across the globe. So what I think we need is a field guide to the microbes to do the same type of thing. And it's very challenging. You've heard many examples of people trying to do this, right, in this series here, but I think it is conceptually possible, and we need to move in that direction. And the way that we need to do this is like the birders. We need a global coordinated effort with lots of people across the globe collecting data, sharing information about samples, identification, abnormalities, normalities, et cetera. And there are lots of examples of people already starting to do this. You've heard examples already here. There's other ones that are going on now that I assume many people have heard about. So there's lots of wastewater surveillance for SARS-CoV-2 and other pathogens that have been ramped up massively in the face of the pandemic. It's been used for many other pathogens. It's been detecting possible animal-derived SARS-CoV-2 in various urban settings. We can mimic this wastewater surveillance to, in theory, do surveillance of water coming out of a cave or surveillance of water coming out of the gorilla areas. I think you could collect samples in a systematic way, build a library, maybe not process them immediately, but you could characterize them for DNA or for other types of information, not just on the microbes, but you can also do things where you collect information for the DNA. There's this whole field called eDNA where people are surveilling lakes and rivers and other systems for skin and DNA from host organisms that you could compile this information for. And how can we do this? We need massive collections to do this. You already heard a little bit of a mention about citizen science, and I think we can have other efforts like this. There's this giant effort that I'm a distant collaborator on called Metasub that has been collecting urban samples from thousands of sites across the globe. We've done a variety of citizen science projects or participatory science projects with scientists and with the public to collect samples, and I think we could do this on a much, much larger scale across the globe in some way that could sample air, water, dust, animals, fecal samples, whatever it is, and really start to have a global field guide to microbes. And I just want to make a point that you've already sort of heard an example of this, but the participation of the public in this also engages the community in thinking about some of the issues associated with microbial sampling. So I just wanted to go really, really quickly because we're running late and turn it back over to the community. But I do think that this is an opportunity to really start to ramp up our game in microbial surveillance.
Thank you, Jonathan, and thank you to all our speakers. We're running a little bit behind our time, but I asked our participants to stick around with us for a little bit of Q&A. A lot of you submitted questions during the registration process, and our panel had a chance to take a look at the questions and summarize them so that we could make better use of our time. So you might not hear your exact question, but you will hear a question reflecting the information that we're hoping to learn from our panel. So I'm going to start with Christine. What regular surveillance activities will be going on to catch pandemic surges as they begin? What do we do right now, and what do you expect we will be doing as technology evolves?
Sure. I mean, I think the future definitely lies, as Jonathan and all the colleagues alluded to, in finding ways where we can have sort of a less intensive effort on the ground, where we can more automate, where we can help scale the type of surveillance that's needed, because we actually need surveillance to sort of be going everywhere. It needs to be, again, at that animal-human interface. We need to combine the two. There's a lot of efforts that are ongoing. I think there was a great question too about federal agencies and what the plans are. And I think, you know, for example, the One Health Institute, we have a lot of programs through DoD and DARPA, as well as USAID to help foster and bring on board other countries and collaborators in surveillance. And I think ultimately that's what's needed, is to have that footprint elsewhere, overseas, where collaborators are doing the work where it matters most, where diseases are most likely to emerge. But this new technology that was described today has a huge role to play in that. Thank you.
Thank you, Christine. The next question is for Christina. And as we talk about going into these wild, maybe more difficult to access environments and the deployment of sensors, is it possible at this point to detect virus in the air using biosensors, and how sensitive is that technology right now?
So that's a great question. It's an area that I'm not actively working on, but others are. And there are groups that are working on sensors that can detect viruses in open air environments. It is really challenging because if you think about the infectivity levels of a virus, the number of particles that you would need to detect to be able to understand that it's a threat or to identify it, it just makes it very challenging to do that detection. So right now, there are not any that are really viable and fielded, but it's an area of active work that different people are attempting to solve.
Thank you, Christina. My next question is for Pranav. We saw in one of your slides a table comparing the number of known hosts for certain viruses versus what is expected to be available as a host for those viruses. So can you talk to us about how confidently can we estimate or predict viral evolution in the face of species jumping from one to another, or other pressures?
Yeah, I think we are probably at a stage where we are not able to forecast, like have a true forecast that someone can use, but we are at a stage where we can grade those viruses, and that risk can be understood in terms of whether this specific virus can have a possibility of cross-species transmission versus a virus that is probably can have host range which is much more narrower. And that also kind of translates something around its ecological tendencies for infecting human beings. So it's at a stage where we are getting confident around our methods. And as more and more data comes along, we will be probably more confident. We'll have more that is, I think the other question that also included was in the questions that were asked was around the modeling challenges. And one of the modeling challenges to have ensemble models that can help us improve the prediction capacities, and those kind of come from different perspectives. So once we have those methodological advances done, we will be more confident around predicting those characteristics for novel viruses.
So hearing everyone talk about the different expertise that is needed in order to make these big leaps forward in the way that we are predicting new pandemics and risks, one of the questions we have is, what kind of skills do we need in our scientist training pipeline? So Chiara, maybe you as a budding epidemiologist, what are we, what classes or what training should people be looking at if they want to make a contribution to this area of research?
Sure, yeah, that's a good question. I think this panel was a really good example that we all come from very different backgrounds in our training, and we're all trying to work together to solve some of these really big questions. And I would say that we all kind of speak slightly different scientific languages when we're thinking about people in engineering and people in veterinary medicine and epidemiology and data science. And so as an early career scientist, taking that time to really invest in taking that extra class that's outside of your field, like pushing your envelope a little bit, or attending that extra seminar that's in a different college than your own, to try to put that effort into learning to slightly speak different languages. So when these big challenges come up, you can work as part of a really diverse team. And then also I would say, learning to and spending time investing in learning to work in different international settings is a huge effort and a really important thing to do. There are several programs that I found very, very valuable for my career that I engaged in as a graduate student, the Fulbright program, which allows you to spend a year working in other countries around the world, or the Fogarty International Center through NIH had the International Scientist Development Award program. And both of those have been excellent programs that I highly recommend.
Thank you. So one final question for everyone, since this is the Team Research Forum. You each in your presentations highlighted challenges or obstacles that you are coming up against as your research progresses. So I would like each one of you to very briefly let me know if you could have your ideal research partner or collaborator kind of jump out in this meeting and say, I want to work there. What skill set or what knowledge are you looking for in a collaborator to help you make that next leap in your research program? Maybe we can start with Christine.
Oh boy, I was going to say it's a whole lot of qualities, but for the most part, I just look for willing and enthusiastic collaboration, especially people who are willing to take the risk of working across disciplines. Invariably, that's going to take more time. I think, as Chiara alluded to, we need to learn each other's ways of working, and what's productive for one of us might not be for the other. So the patience and the willingness to leap over those boundaries is what I look for. Thanks.
I think great question for me as well. So one of the things that I've looked for over time is people who are just willing to talk, and that goes a long way. And maybe something really exciting will work out, and maybe next time it's a different conversation. But just people being willing to have those conversations is really fantastic and very helpful. And then also understanding that timelines in different disciplines operate a little differently. And so some things that seem easy from the outside actually are really challenging because we don't know all of the little, things that can go wrong or go right. And so just, again, those communication skills are really helpful in these big interdisciplinary teams.
Jonathan?
People who can engage and leverage the entire population of the planet in terms of citizen science and participatory projects, and getting people, like cell phone providers and UPS and airplane companies and others, because we could actually have a really interesting global collection of data if we engaged people more.
Pranav?
Yeah, I think I kind of agree with what Chris Johnson was saying. Along with someone who is willing to learn new techniques and new concepts, I think I would like an ideal collaborator for me would be also someone who is willing to take risks, and because these are very challenging problems that we're trying to address. So someone who's ready to take and handle those challenges is what one of the criteria, I would say.
Who am I missing?
I think I'm last. Yeah, I echo everything that all the colleagues have said here. Great communication skills, willing to think outside of the box and be highly creative. And at the moment, someone who's willing to help me get something to the top of those gibbon trees, like a breathalyzer, would be really, really fascinating.
I think I'm the last one. I think the skill set I'm looking for is kind of more specific than my colleagues. So I don't know whether we have a lot of students or postdocs in the audience here. So I would say one is coding, and the other one is hands-on skills. So basically, they can not only code the code, but then they can actually to test this with some robots actually, and then they are not afraid to fail because we are going to crash a lot of drones or the other robots. So that's the 3 skills that I'm looking for.
That's perfect, actually, Zandan. The willingness to take risks and to just proceed anyway in the face of failure, I think, is something that's probably more important for success than we realize. Very well said.
I want to thank again our panel for a wonderful presentation and for your insights into this area of research and encouraging us to look into it as a potential avenue for many of our researchers, postdocs, and students. This session is recorded, so we'll be able to share it with everyone. And if you are interested in learning a little bit more about this team's group and upcoming opportunities to engage with them or events, please feel free to contact Professor Christine Johnson directly or reach out to me or our team at Strategic Initiatives, and we will be more than happy to help you make that connection. I want to thank everyone again for a wonderful session. Wish you a great weekend, and we will see you in a couple of weeks for a session on bio-EMS with the College of Biological Sciences. Thank you again. A big hand to our panel. Have a wonderful weekend.
Thank you all.